Hey guys, this video is about acid-fast stain or Gill-Nelson stain, which is also known as Jatan stain. Jatan stain was first introduced by Paul Ehrlich and modified by Frank Gill and Friedrich Nelson. It is a type of differential stain that imparts color to the acid-fast organism. Here, acid fastness is attributed by the cell wall with high waxy content of lipid and fats as well as higher alcohol group, and acid-fast organisms are those which are capable of retaining the primary stain when treated with acid. And mycolic acid is usually present in all of the acid-fast organism. We need to prepare a thin smear and heat fix it by passing slide 3 to 4 times through the flame, and then we need to put the smear over the rack and pour carbylphosphine on the slide, and then we need to gently heat the slide from underneath by the means of spirit lamp until slight steam comes out, and leave it for 10 minutes. Just remember, we don't need to boil the smear, and this process of gentle heat helps stain to penetrate through the bacterial cell wall. After 10 minutes, we will rinse the smear with water, and then we will add the decolorizer, which is 20% sulfuric acid, for about 30 seconds to 1 minute. However, for Microbacterium leprae, we'll use 5% sulfuric acid, and for the bacterial spores, we'll use 0.5% of sulfuric acid. Then we will finally rinse with water, and the last step, that is counter stain, we will do that by 2% methylene blue or malachite green for 2 minutes. Then after the final step, we will rinse the smear with water and visualize microscopically. Finally, under the microscopy, we will find that acid-fast organism will appear red color and non-acid-fast organism will appear blue color. So here is a stained smear of AFP positive organism. So the major step after the microscopy is reporting of the slide, which is very important. Now we will do the reporting as follows. If we see 0 AFP per 100 field, then we will report it as no AFP seen. If there is 1 to 2 acid-fast bacilli per 300 field, we will report it as scanty. If 1 to 10 AFP per 100 field, then 1 plus. If 11 to 100 AFP per 100 field, then 2 plus. If 1 to 10 AFP is seen per field, then we will report it as 3 plus. And if there is more than 10 AFP per field, then we will report it as 4 plus. So this was about the steps and reporting of Jatan staining. The basic example of acid-fast organism is the first one, most importantly, Microbacterium tuberculosis, which causes TB, and then Microbacterium leprae, Nocardia species, spores of Cryptosporandium, etc. This was all about the Jatan staining procedure. Thank you.